Welcome to Studious. Today, we are exploring one of the most underappreciated yet utterly essential structures in the entire central nervous system, the choroid plexus. Hidden deep within the fluid-filled cavities of the brain, called ventricles, the choroid plexus is the primary site of cerebrospinal fluid synthesis, and it serves as the guardian of the brain's internal chemical environment. Without the choroid plexus working continuously, 24 hours a day, 7 days a week, the brain would be starved of nutrients, flooded with toxic metabolic waste, and left vulnerable to physical trauma. The choroid plexus is present in all 4 ventricles of the brain: the 2 lateral ventricles in the cerebral hemispheres, the 3rd ventricle near the thalamus, and the 4th ventricle at the base of the brain near the cerebellum, each contributing to the continuous flow of cerebrospinal fluid. In this video, we will map its location, examine its microscopic structure, trace the mechanics of fluid synthesis, follow the complete circulation pathway, and understand every function CSF performs.
Before we examine the choroid plexus in detail, let us first understand precisely where it sits within the architecture of the brain. The choroid plexus occupies the innermost cavities of the brain, the ventricular system, positioned deep within the forebrain and midbrain, perfectly protected by the surrounding cerebral structures. When you look at a mid-sagittal brain section, the choroid plexus appears as a delicate frond-like green tissue lining the roof of the ventricles. Above it sits the massive corpus callosum, the great white matter commissure connecting the 2 cerebral hemispheres. The cerebral cortex forms the outer wall, and the thalamus and its interthalamic adhesion sit immediately posterior and lateral to the 3rd ventricle. The fornix, the limbic system's memory highway, arches directly above the choroid plexus in the lateral ventricles. This deeply protected location is not accidental. The brain has placed its fluid-producing engine in its most sheltered, central space, insulated from external trauma, with direct access to the arterial blood supply it needs to synthesize cerebrospinal fluid continuously.
Now let us zoom into what the choroid plexus actually is structurally. The choroid plexus is an invagination of the pia mater, the innermost of the 3 meningeal layers surrounding the brain. Invagination means a folding inward. The pia mater literally folds into the ventricle, bringing its blood supply with it, creating the highly vascularized tissue we call the choroid plexus. Under magnification, the choroid plexus has an appearance similar to a richly branching cauliflower or sea coral. This folded, frond-like morphology dramatically increases the surface area available for fluid production. Its structure consists of 3 layers. The outer covering is a continuous sheet of specialized ependymal cells called choroid epithelial cells. Beneath this epithelial layer lies a richly vascularized connective tissue core, a dense network of blood vessels and capillaries derived from the pia mater. The entire tissue is richly innervated, with autonomic nerve fibers modulating its secretory activity. The primary function of this entire structure is continuous, active production, synthesis, and circulation of cerebrospinal fluid into the ventricular cavities.
The choroid plexus is not confined to one location. It is distributed across all 4 ventricles of the brain simultaneously, though in different quantities and with different output volumes. In the lateral ventricles, the paired C-shaped cavities in the cerebral forebrain, one in each hemisphere, the choroid plexus is at its largest and most productive. These are the primary sites of CSF synthesis, producing the highest volume. CSF exits the lateral ventricles through the interventricular foramina, also called the foramina of Monro, 2 narrow channels connecting each lateral ventricle to the 3rd ventricle. In the 3rd ventricle, the central narrow cavity adjacent to the thalamus, a smaller choroid plexus contributes secondary CSF volume. CSF exits here via the cerebral aqueduct, also called the aqueduct of Sylvius, a narrow channel running through the midbrain. In the 4th ventricle, located between the brainstem and the cerebellum in the lower brain, another choroid plexus adds further secondary contribution. The critical insight here is that all four choroid plexuses are actively synthesizing CSF simultaneously, creating a continuous, uninterrupted production system throughout the entire ventricular network.
To truly understand how the choroid plexus produces cerebrospinal fluid, we must examine its microscopic architecture, the two distinct layers that make it both a fluid factory and a precision barrier. The first layer, the core, is composed of highly vascularized connective tissue derived from the pia mater. This core is packed with a dense network of fenestrated capillaries, blood vessels with small pores in their walls that allow plasma to leak into the surrounding connective tissue relatively freely. This leaky vascular core provides the raw material, blood plasma that is filtered out of the capillaries into the connective tissue space. The second layer, the cover, is a single sheet of simple cuboidal epithelial cells called choroid epithelial cells, which sit on a basement membrane and form the outer boundary of the choroid plexus, facing the ventricular cavity. Critically, these epithelial cells are joined by tight junctions, molecular seals that prevent anything from passing between cells. This epithelial layer is continuous with the ependyma, the glial cell lining of the entire ventricular system, creating one unbroken cellular boundary between the blood system and the cerebrospinal fluid. The choroid epithelium is therefore both the factory floor and the quality control checkpoint of CSF production.
Now let us trace the precise three-step mechanism by which the choroid plexus converts arterial blood into cerebrospinal fluid. Step 1. The source. Arterial blood, supplied by branches of the anterior and posterior choroidal arteries, enters the highly vascularized connective tissue core of the choroid plexus. Because the capillaries here are fenestrated, meaning they have pores, blood plasma filters freely out of the capillaries into the surrounding connective tissue stroma. This plasma ultrafiltrate is the raw starting material for CSF. Step 2. The engine. This plasma ultrafiltrate now encounters the choroid epithelial cell layer. Fluid crosses this layer through two concurrent processes. First, passive diffusion. Water and small dissolved molecules move across the cell membrane down their concentration gradients. Second, and more importantly, active transfer. Specialized ion transport proteins embedded in the epithelial cell membranes actively pump specific ions, particularly sodium, chloride, and bicarbonate, from the plasma side into the ventricular cavity side. Water follows these ions osmotically. Step 3. The output. The result emerging on the ventricular side is pure, precisely formulated cerebrospinal fluid, secreted continuously into the ventricles at approximately 500 milliliters per day in an adult.
One of the most important functions of the choroid plexus epithelium is its role as the blood-CSF barrier, a highly selective molecular gatekeeper that strictly separates the systemic blood circulation from the cerebrospinal fluid environment of the brain. The blood-CSF barrier is distinct from the more widely known blood-brain barrier, which is formed by the tight junctions of cerebral capillary endothelial cells. In the choroid plexus, the barrier is formed instead by the tight junctions between adjacent choroid epithelial cells, which prevent any paracellular leakage, meaning nothing can slip between the cells without being screened. The barrier performs two critical roles. First, ionic homeostasis. By acting as a selectively permeable barrier, it precisely controls the movement of specific ions, nutrients, and other substances from the blood side into the CSF, maintaining the exact ionic composition the brain requires. Second, developmental importance. Maintaining this stable, tightly controlled internal environment is absolutely critical for the central nervous system, particularly during early fetal brain development, when the choroid plexus is one of the very first brain structures to form and begin functioning, providing the developing neurons with their precisely controlled chemical environment before the blood-brain barrier has fully matured.
Once cerebrospinal fluid has been synthesized and secreted into the lateral ventricles, it begins a precisely directed journey through the entire ventricular system, a 5-step internal transit pathway before it exits the brain entirely. Step 1. Secretion. CSF originates in the lateral ventricles, produced by the choroid plexus located in the body and inferior horn of each lateral ventricle. Step 2. Transit. CSF flows medially and inferiorly through the interventricular foramina of Monro, two narrow, paired openings connecting each lateral ventricle to the midline third ventricle. These foramina are clinically important because a tumor or cyst blocking them causes obstructive hydrocephalus, dangerous CSF buildup and pressure elevation. Step 3. Arrival. CSF enters the third ventricle, where it mixes with the smaller CSF contribution from the third ventricle's own choroid plexus. Step 4. Transit. CSF then flows inferiorly through the cerebral aqueduct, the narrowest segment of the entire CSF pathway, running through the midbrain. Obstruction here is the most common cause of non-communicating hydrocephalus. Step 5. Terminal internal station. CSF arrives in the fourth ventricle, the final ventricular cavity, where it receives the fourth ventricle choroid plexus contribution before preparing to exit the ventricular system entirely.
The internal transit through the ventricles is only part of the CSF journey. After reaching the fourth ventricle, cerebrospinal fluid exits the closed ventricular system and begins circulating around the exterior of the brain and spinal cord in 3 final stages. Step 6. The exit. CSF leaves the fourth ventricle through 3 distinct apertures. 2 lateral apertures, called the foramina of Luschka, located on each side of the fourth ventricle, and 1 median aperture, called the foramina of Magendie, located in the midline at the inferior end of the fourth ventricle. Through these 3 openings, CSF enters the subarachnoid space, the fluid-filled compartment between the arachnoid mater and the pia mater that surrounds the entire brain and spinal cord. Step 7. The outer path. Once in the subarachnoid space, CSF flows superiorly, upward over the surface of the brain and down along the spinal cord, bathing the exterior of the entire central nervous system. Step 8. Reabsorption. CSF is ultimately reabsorbed at the superior surface of the brain, where specialized projections of the arachnoid mater, called arachnoid villi or arachnoid granulations, project into the dural venous sinuses and actively transfer CSF back into the venous blood system, completing the cycle. The entire CSF volume of approximately 150 milliliters is replaced roughly 3 to 4 times per day.
Cerebrospinal fluid is not simply diluted blood or a generic saline solution. It is a precisely formulated biological fluid with a specific chemical composition crafted by the choroid plexus to meet the exact metabolic needs of brain tissue. Its composition can be understood in 3 layers. The base element is water. CSF is approximately 99% water, making it a clear, colorless fluid with a consistency similar to distilled water. This is why normal CSF appears crystal clear on lumbar puncture. Any cloudiness, blood staining, or discoloration is immediately clinically significant. The regulators are specific plasma components, including sodium, potassium, calcium, magnesium, chloride, and bicarbonate ions, maintained at tightly controlled concentrations that differ significantly from blood plasma, reflecting the choroid plexus's active selective transport rather than passive filtration. The fuel consists of high concentrations of amino acids and glucose, specifically formulated to continuously nourish the delicate neurons and glial cells of brain tissue. Importantly, normal CSF contains very little protein and essentially no red blood cells. Their presence in CSF analysis is a key diagnostic finding in meningitis, subarachnoid hemorrhage, and other neurological emergencies assessed during lumbar puncture.
Having understood how CSF is produced and what it contains, let us now map the 4 critical systemic functions that cerebrospinal fluid performs for the brain and entire central nervous system. First, nourishment. CSF feeds delicate brain and spinal cord tissue through its continuous delivery of amino acids, glucose, and essential ions. This is particularly vital in brain regions where direct capillary supply is limited and where neurons depend on CSF as a supplementary nutrient reservoir. Second, shock absorbance. CSF suspends the brain within the subarachnoid space, effectively making the brain float in fluid. This hydraulic cushioning provides a critical mechanical buffer against mild to severe head trauma. Without CSF, even moderate head impacts would translate directly into brain tissue, causing catastrophic neuronal injury. Third, waste removal. The brain is unique among body organs because it entirely lacks a lymphatic drainage system. CSF, therefore, serves as the brain's functional equivalent of the lymphatic system, continuously flushing metabolic waste products, including excess neurotransmitters, proteins, and cellular debris, out of the central nervous system through its reabsorption cycle. Fourth, environmental homeostasis. CSF maintains the strict ionic balance and chemical stability required for optimal neuronal firing, synaptic transmission, and overall neural function.
We close with the grand synthesis. The choroid plexus is not merely a biological faucet passively dripping fluid into the brain. It is a master regulator of the brain's entire fluid ecosystem, performing 3 integrated roles simultaneously. As the synthesizer, it relentlessly converts arterial blood into the vital, nutrient-rich lifeblood of the central nervous system, producing 500 milliliters of precisely formulated CSF every single day. As the circulator, it drives a continuous mechanical flow through the ventricular system, through the subarachnoid space, and back into venous drainage, a circuit that nourishes tissue, removes waste, and compensates for the brain's complete absence of lymphatic vessels. And as the protector, it stands as an impenetrable gatekeeper through the blood-CSF barrier, isolating the brain's fragile internal environment from the chemical chaos of systemic blood flow, ensuring that the neurons responsible for every thought, movement, memory, and emotion you experience are bathed in a perfectly stable, precisely controlled environment. Pathology of the choroid plexus, from tumors called choroid plexus papillomas producing excess CSF, to inflammatory damage disrupting the barrier, has severe neurological consequences. Understanding the choroid plexus is understanding the foundation of brain stability itself. Thank you for watching Med Studios.